and their genome available. The database is available, so you can explore it and correlate it with human beings. The human genome is already there, right? So this is another important aspect that I wanted to focus on. Now, we'll start with the first one. I deliberately skipped the prokaryotes, say E. coli or Bacillus subtilis. I thought that I will start with, because I do not want you to bombard with a lot of information. So I started with the yeast, that is Saccharomyces cerevisiae. Now this is an electron micrograph with the work of Ramasamy and Dasmurthy, and it shows the image of Saccharomyces cerevisiae, which is also commonly called as the baker's yeast. Now, if you come across Saccharomyces cerevisiae, you would again wonder why this is called as or why this is referred to as the model organism. And this is one of those model organisms which is unicellular. So there are some microbiologists also over here who would know that it is rapid to grow under laboratory conditions, and it can easily be cultured. So this yeast can easily be cultured in the laboratory. It is unicellular. Then two DNA transformation studies can also be carried out easily in S. cerevisiae, and it has a small eukaryotic genome. Now, as compared to the Bacillus subtilis or E. coli, it has a small genome, and it becomes more important because it is eukaryotic species, right? So though unicellular, it is eukaryotic and has a small genome. And how small? So you can see it is around, say, 12 lakh, 1 crore, or say that much base pairs, and 5,770 genes. And if you compare this with human beings, it is around 3 into 10 to the power 9 base pairs, and more than 20,000 genes in human beings. So it becomes comparatively easier for us to carry out studies on Saccharomyces cerevisiae. Important significant distinguishing feature of Saccharomyces cerevisiae is that it can be present in both haploid as well as diploid state. If you look at the reproductive cycle of Saccharomyces cerevisiae, you will see that it can be in haploid state as well as in diploid state. And it being in haploid state, it can be specifically helpful in the studies where recessive mutations are at risk. You know, recessive mutations, if the condition is diploid, it will be difficult for us to study the recessive mutations because a dominant character may come in, right? So when there are recessive mutations, haploid conditions makes it easier to help them to study, right? So haploid state of Saccharomyces cerevisiae makes that easy. Now, and then once you have studied the haploid forms, the complementation test can be carried out in the diploid strains and can be correlated in the same way. This is something unique with Saccharomyces cerevisiae. Another thing is the exogenous DNA can be incorporated using recombination. So like I had mentioned in the earlier slides, that recombination, or those organisms which can undergo recombinations, can be good model organisms as well. So you can use yeast as a tool for expression system of heterologous proteins also. And there are several studies which have been carried out on Saccharomyces cerevisiae with respect to one important aspect, that is aging. Now, when we say how high throughput method for quantitative measurement, we mean that can we carry out an experiment at larger scale with a large number of molecules being tested in one set, maybe. That is called as high throughput method for quantitative measurement of chronological lifespan. So Murakami and Kebelin in 2009 have identified some markers which are common in Saccharomyces cerevisiae and complex organisms like vertebrates also. So aging can further be correlated from Saccharomyces to vertebrates and especially human beings. The work of Biddick and Young 2009 shows that gene expression regulation, how it is carried out in Saccharomyces cerevisiae. Like I was addressing in the initial slides, that this Baker's yeast is an important organism where signaling pathways are studied. So signaling pathways and regulatory processes associated with osmoregulations have been studied by Homan et al. In 2007. Cell cycle we all know, and its regulation has been studied by Heinz-Peter Noscher and his co-workers in 2002. Several metabolic pathways have been studied by Brockett Mason and Bruno Dumas 2006. Apoptosis has been studied in several model organisms, right, till zebrafish. And Saccharomyces cerevisiae is no exception, where Osianowski has studied this along with his co-workers in 2008. Neurodegenerative disorders such as Alzheimer's have been studied by Miller Fleming et al. In 2008, and so on. So now, having so much of study going on, these are some of the works which have been carried out in 2000, early 2000 only. So the choice of Saccharomyces as the model organism has done one thing. It has made yeast as a big winner. Winner in terms of what? What do you normally associate when an organism is constantly being worked upon, and then on your eyes a news breaks up? A big winner in terms of Nobel Prizes. So you see, Saccharomyces cerevisiae, or the baker's yeast, has won a lot of Nobel Prizes for finding the key regulators of the cell cycle. Hartwell, Hunt, and Nurse in 2001 were awarded Nobel Prize in Physiology or Medicine. Right now, I'll make a point over here that it does not mean that they published some work in 2001. If you go through the literature, the work which Hartwell, Nurse, and Hunt carried out, it dates back well into the 70s and 80s, and for which they get recognized quite late in 2001, right? So it is not sometime immediately that you get a Nobel Prize. It is sometime recognized pretty late. So key regulators of the cell cycle was the area we read it in, Cooper and Karp and Bruce Alberts. Those observations were the ones made by these people for through Saccharomyces cerevisiae, for which they got Nobel Prize. Then we have heard this name Kornberg along with another Indian name, if you can recall. Can somebody tell me? Kornberg has been associated with one American-based Indian who got the Nobel Prize for synthesis of DNA. Can somebody tell me? The name was Hargobind.
Hargobind Khorana.
Right. Happy to hear that. Hargobind Khurana. Right. So Kornberg 2006 got this Nobel Prize in Chemistry for depicting the molecular basis of eukaryotic transcription. Now, when I'm talking all this, try to recall the things that you have studied in your MSc classes. How transcription takes place in eukaryotes, how transcription takes place in prokaryote. So all those mechanisms that you have studied that regulates the eukaryotic transcriptions have been studied by Kornberg, and those studies were carried out again on Saccharomyces cerevisiae. Now these two ladies and one gentleman, Blackburn, Grieder, and Zostak, they got their Nobel Prize in 2009 for their work on telomeres and enzyme telomerase. Now this telomeres and telomerase, the enzyme, these are interestingly associated a lot with aging. One of my friends, Amruta Bhusari, works in Ohio State. She also works on telomeres and telomerase enzyme, which is an interesting aspect. It says that the ends of chromosomes, they degrade, they degenerate with every cell division, right? And that probably leads to the aging. And they have worked on this aspect of telomeres and telomerase in Saccharomyces cerevisiae. These three gentlemen, Rothman, Schickman, and Sodov, in 2013, they got the Nobel Prize in Physiology and or Medicine again for the transport system in cells. If you can remember how the membrane system functions in the cell with respect to the vesicle transport from, say, endoplasmic reticulum to Golgi complex, then Golgi complex vesicle formation to various parts of the cell, or maybe exocytosis and all. So they had their findings on transport system in cells specifically carried out in Saccharomyces cerevisiae. And so you can see 1, 2, 3, and fourth Nobel Prize for the works on Saccharomyces cerevisiae. And then recently in 2016, Oshumi from Japan, he got a Nobel Prize in the field of physiology or medicine for looking into the aspect of autophagy. Autophagy means self-eating. And here he has seen Saccharomyces as a model organism which correlates the finding of how human cells destroy themselves. That means autophagy, right? So you would see that Saccharomyces cerevisiae has earned as many as 5 Nobel Prizes, right, for several people. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and 11. So 11 people could get Nobel Prizes just because of Saccharomyces cerevisiae. So I do not know if some of you would be interested in getting Saccharomyces cerevisiae and start your work, hoping that you get a Nobel Prize also. I don't know. The second interesting one is the worm C. elegans, right? And after talking about C. elegans, maybe we'll take a small break and start again, because I don't think my talk will be lasting for 2 hours, 3 hours maybe. Maybe we'll finish by 2.5 hours, so maybe 2 hours. So that's why I'm going very gradually. I do not want to give a lot of information today as such, but at the same time want to fetch your interest on how if you can choose one of these as the model organism, or at least know some of the model organisms or the aspect associated with that. So second interesting organism, which is a worm, is nematode worm, is C. elegans, right? Caenorhabditis elegans, right? So if you just look at this microscopic image of C. elegans, you would see that it is quite simple, very simple structure. C. elegans, easy to grow. You can have some culture medium in the laboratory and grow it. C. elegans, short lifespan, as I had already mentioned, few 3.5 days or so, short lifespan. And a lot of genetic studies can be conducted in C. elegans in both ways. One, self-fertile, that means cross-fertilization can take place, and self-fertilization can also take place. And like zebrafish, because it is transparent, you can look under the microscope and see several changes occurring in the organism itself. And it is a small size, so you never wonder actually how to maintain these cultures in the laboratory. So size is never a problem. And it has an invariant cell lineage, fully described anatomy and development. So a lot of information is already available. Like I said, it is one of the organisms for which the genome sequencing was carried out in late 90s and early 2000. So the complete genome is also sequenced. And one of the important work that has been carried out, for which a Nobel Prize could also be won through C. elegans, is RNAi discovery, right? So RNAi knockdowns can also be performed. These can be frozen and preserved, and you do not need expensive infrastructures. So no animal house is needed. You can easily maintain them in the laboratory. They do not pose any biohazard. Imagine culturing a cockroach in the laboratory. So if you want to culture a cockroach, or say for example earthworm or something else, then there are so many other problems associated, that sometimes they act as a pest, sometimes they attract some other organisms also. But with C. elegans there is no such problem, and you get a big size of, like I said, In a day, they give around 140 eggs. So the number of progeny is also very high, and therefore C. elegans can be a wonderful choice of model organism for your research work also. So Brenner was somebody who was more interested into C. elegans, and ever since Brenner found this and started working on C. elegans in 1963, there were a lot of mutants which were observed by Brenner in C. elegans also, and those were published in 1974. And Sulston and Howitz in 1976, they determined some of the post-embryonic cell lineages as well, which become one of the aspect of research for many. Howitz in 1982 studied programmed cell death, that is apoptosis, in C. elegans. Then complete embryonic cell lineages were also determined by 1983 by Depet and his co-workers. Connectome. Now, connectome is I would say, the wiring connections of the nervous system. So complete connections of the nervous systems were established in C. elegans, and it comes as a really good model in understanding the nervous systems. So that was done in 1986. Then I was talking about interference RNA, RNAi, and miRNA. They were discovered in C. elegans in 1991 to 1998, for which Fire and Mello got Nobel Prize in 2006. Again, this green fluorescent protein was firstly used in C. elegans only in 1994, for which Chaffee was awarded Nobel Prize in 2008. This was the first animal whose complete genome was sequenced, as we have seen in 1996 or so. So 97 MB, which was then sequenced, and now we know that it is 100.3 MB of genome. And we saw that Saccharomyces cerevisiae has around 5 Nobel Prize won for his work, for its workers, right? And C. elegans is not far behind. So Brenner, like the name I mentioned, started somewhere around 1963, along with Howitz and Sulston, got the Nobel Prize in Physiology or Medicine in 2002 for their studies on genetic regulation of organ development and programmed cell death. Fire and Mello, as I mentioned just now, they got their Nobel Prize for Physiology or Medicine for the discovery of RNAi in C. elegans. They got this Nobel Prize in 2006. And then we have the Nobel Prize in Chemistry in 2008 for the application of GFP, green fluorescent protein, for the first time. Shimomura, Chaffee, and Roger. Now, this GFP has become a very common tool even in Drosophila now. So it was these Shimomura and his workers, Chaffee and Roger, who started this first in C. elegans and now has become reasonably common. So till now, what we have seen is why would we look into some organisms as model organisms, number one. Number two, how do I ascertain that a given organism is a model organism? What are the characteristic features that an organism should have to be called as a model organism? Number two, that is what we have seen. Number three, we saw the model organisms with respect to their lifespan, their fecundity, then their evolutionary aspect, and the genome sequencing also. We have also seen some of the model organisms, for example, Saccharomyces and C. elegans. And now we are looking into the plant Arabidopsis thaliana, which is also called the mustard plant. And why this Arabidopsis becomes a model organism, especially in botany, is because of its short life cycle. From germination to mature seed, it takes around 48 hours only. So it is again quite a small time for us to look into the reproductive cycle from the aspect of the plant. As I said, it is from the mustard family. It can be grown in small spaces. You can also grow it indoor. That means in laboratories also. So what it requires is nothing but some soil, water, and light. That's it. So you do not have much of requirements also. And like you have all seen the pods of a mustard plant, a large number of seeds can be obtained from a single pod. So that is also one of the benefits to be treated as a model organism. Compare this with some other tree or some other plant where lesser number of seeds would be obtained. And you can easily cross this with other varieties as well. Mutational screenings can also be carried out, and as compared to, say, for example, Z maize and Oryza sativa, it has comparatively smaller genome, which is, say, around 132 megabase pairs, and was sequenced in 2000, right? And it has two major seed stock centers also available. This we'll be discussing with respect to Drosophila also. So when you are, looking into a model organism, you are always looking for authentic variety or species of the organism, which you would procure probably from some stock centers or repositories. So there are two major seed stock centers available. I'm not sure where they are, but you can easily find out one. And transformation experiments have worked with Agrobacterium tumefaciens also. So because it has come from Agrobacterium tumefaciens, tDNA is something what it is called as. Now the work of, in fact, the paper of Provat and his co-workers mentions that there are several studies which have been carried out Arabidopsis for the first time in any plant, right? So it was first plant to get its genome sequenced, and like I mentioned, tDNA collections were there for the first time in Arabidopsis thaliana. Then live cell imaging of cellulose synthase was also done for the first time in A. thaliana. Optical detection of hormones and metabolites. I was addressing that a lot of work in Arabidopsis has been carried out with respect to the plant hormones. So optical detection of the hormones and metabolites were carried out for the first time in this plant. Then transcriptomes of entire plant organ, cell type specific transcriptomes, they were studied. And floral ABC models, first genetic and molecular tests were carried out. Hormone pathways were deduced. And molecular glue model with respect to auxins and similar other phytohormones were also studied in Arabidopsis. Plant immune receptor gene was for the first time isolated from Arabidopsis. The idea of basal defense, it came into existence from the studies carried out on Arabidopsis. Cryptochrome, phototropin, UVB, and other receptors, they were identified for the first time in Arabidopsis. The transporters, amino acid transporters, iron transporters, they were studied. Then several similar such works have been carried out for the first time in Arabidopsis only. So like we have seen so many model organisms in animal kingdom, Arabidopsis is doing a similar job in plant kingdom also. Then we come down to this Denio, which is one of the vertebrate model I have chosen for the day. It's a small fish. Small fish you can easily keep in, say for example, आपल्या घरी एक पातेलं असतं त्याच्यामध्ये सुद्धा तुम्ही त्याला रेअर करू शकता, कल्चर करू शकता. इतका लहान मासा आहे Denio, and because of those stripes you can see, this is also commonly called as a zebra fish, right? So Denio or zebrafish, it shares about 70% of genes which are critical with respect to the pathways for growth and other aspect. And these genes remain highly conserved. When we say remains conserved, that means the sequence are similar in other species also. So because of its genetic makeup, it is big choice as a model organism. And the embryo that come out of, they hatch out of the eggs in zebrafish, they are transparent. So it is easy for one to observe the developmental stages and developmental changes in these embryos real time, even at the cellular level as well. And it takes around say 72 hours only after the fertilization for the development of important organs, vital organs, as well as the major part of the nervous system, that is the central nervous system also. So again, you can quickly observe the changes and the effect in course of the research that you are carrying out. And it does not cost much. Like I was saying, if I'm working on labia or katla or some other carp or some other fish, it will cost me hundreds of rupees per kg of fish. And if I'm doing the experiments repeatedly, it will be cumbersome for my pocket also. So zebra fishes are comparatively cheaper because, the lesser weight, small size, you will get big number of zebrafish in the same cost as compared to the other animal models. And initially, when we were talking about, say, Saccharomyces cerevisiae, I was talking about high throughput sequence screening. And the meaning here again is that you are studying the effect of large number of molecule in one experiment, or say or so. So zebrafish models, they are good at that as well. They have been I would say, experimental or mm, good with the gene editing systems such as Cas9 and CRISPR for editing the zebrafish genome. And next generation NGS DNA sequencing has also worked well with the zebrafish. So zebrafish these days is another good choice as a model organism. A lot of work has been carried out on D. rerio, zebrafish, right? So I have enumerated some of the works with the links to these research papers also, or these studies also. So a lot of work has been carried out on zebrafish with respect to toxicology, where the toxicology studies with respect to the microplastic and nanoplastic toxicity. It has been recently studied on zebrafish. Then uh, this actually has seen zebrafish as the model for the toxicological studies and drug efficacy studies also. Then zebrafish has emerged as the model organism for the studies with respect to metabolic disorders as well. Along with that, it has therefore established itself as a biomedical model. There are studies with respect to the obesity also. And so this paper I found really interesting, where the small fish can help us to study the aspects and the reasons, underlying reasons for the obesity in human beings also. So one would wonder, a small fish like zebrafish and elucidating the mechanism, underlying mechanism, what causes the obesity in human beings as well. So the metabolic disorders have been an area of interest with zebrafish. Then there is the aspect of bone disease and related studies also, where zebrafish model have been studied with respect to the human skeletal disorders also. Some studies related to osteoporosis and skeletal biology in human beings as well. Then cancer related drug delivery and drug discovery studies have been also carried out in zebrafishes, along with correlation of these studies to human cancer and some non-mammalian vertebrates as well. Some precision cancer therapy study have been carried out in zebrafish as well. And like I have been repeatedly telling you, that neurodevelopmental disorders have found important space in all model organisms, such as the Parkinson's disease and Alzheimer's disease. And like I said, I'll be talking about one interesting aspect later on when we talk about Drosophila. So zebrafish therefore has found spaces in several aspects, whether it is toxicology, whether it is metabolism, whether it is skeletal system, or neurodegenerative diseases, and cancer as well. That brings us to the last part of today's presentation, which talks about Drosophila melanogaster. So out of all these, I thought Drosophila can be an organism of interest. One, because you do not need to go anywhere to find Drosophila, right? Or zebrafish, maybe you will ask some supplier to provide to you. Then C. elegans, probably you'll need to set up a culture, or you'll look for a repository somewhere. Drosophila, you can easily get, catch hold of anywhere. Even यांना मराठीमध्ये आपण चाचण म्हणतो किंवा चाचणी म्हणतो. The only thing is that you should be sure that this is Drosophila melanogaster only, and it is not some other species like Drosophila suzuki. There is another species, Drosophila suzuki, right? So the common name of this dipteran insect, this is an insect, phylum Arthropoda, invertebrate animal, and the common name is fruit fly. And those who are not from the zoological background would recall that whenever there are some ripened fruits in your kitchen, आपल्या किचन मध्ये अनेक वेळा काही फळं पडलेली असतात, ती खूप पिकलेली असतात, स्पेशली पेरू असेल किंवा केळी असतील, तर त्याच्यावर आपल्याला काही बारीक माशा फिरताना दिसतात. अनेक वेळा आपण ड्राईव्ह करत असतो तेव्हाही आपल्या डोळ्यासमोर काही माशा येत असतात, बारीक बारीक माशा येत असतात. मोर ऑर लेस अनेक वेळा त्या या माशा असतात, विच आर कॉल्ड एज फ्रूट फ्लाय, ड्रॉसोफिला मेलॅनोगॅस्टर. अँड एज आय सेड, you have to be really careful in identifying this species, whether it is Drosophila melanogaster or some other species of Drosophila. So आपण generally repository मधून ते घेतो, we take it from the repository. The reason is that if you do not purchase this or buy this from the repository or fly facility, then there is a risk you have probably taken up some wrong species. Choosing wrong species can also be really harmful for your research. I'll just, because we have some time also, and you all are doctoral students, you should be really aware of it. I remember my lab mate, one of my lab mate, who was supposedly working on Musca domestica, the common housefly, and she was looking into the aspect of radiation and radiation related studies on Musca domestica. And after say 5 or 6 years of her work, one fine day when she was presenting her work, she found that her guide and somebody who supplied the flies to her said that this is not Musca domestica. In fact, it is stable fly. Stable fly means that bigger mashi, bigger fly that you see in the stables. Stables means jahan par ghode rakhe rehte hain wahan par. Right. So now the 6 or 6.5 years of her work went into vain, because she was looking into all aspects from the perspective of Musca domestica, the common housefly. The same thing happened with me also when I was working under ex-vice chancellor of Goa University, Dr. Dilip Devbakar, and I was supposedly working on two bacteria, Cellulomonas and Azotobacter, and I was giving my interview for senior research fellow SRF, and the expert who came for that interview, he said that, I mean, sarcastically, he said that, Srihari, if these are your observations, then your paper should be coming in Nature. I understood his sarcasm and I said, why are you saying so? What you are claiming as Azotobacter is not actually Azotobacter. I said, sir, I have taken it from NCL, and so far as I know and the work I have done, there is no contamination that I have brought into it. But no, you have contaminated the cultures. I said, fine, then we'll get culture again from NCL and perform the same set of biochemical identification and gram staining, etc. So I did the same set of experiments again and showed it back to my guide, only to realize that it actually was not Azotobacter at NCL. There was a contamination at NCL, the repository at NCL, National Chemical Laboratory, Pune. And that was when my guide said that let us restart the whole thing around and get the culture from IMTech Chandigarh, the Institute of Microbial Technology Chandigarh, and start back afresh. I said, no, I mean, 5 years again down the time, that was a period when I got frustrated. So choice of your organism and identifying your organism correctly is very important. This is the point that I wanted to make, just because that came into my mind. So good thing with Drosophila melanogaster is that it is cosmopolitan distribution. So no need to worry whether you are in Maharashtra, whether you are in Tamil Nadu, whether you are in Northeast, wherever you are working, you are bound to get Drosophila. So even in America or any other place across the globe, you will get Drosophila, right? Because the temperature conditions under which it works is wonderful. very good temperature conditions. 25 degrees good for Drosophila. So you'll find it anywhere. And as I said, it can be collected from nature. So even now, this evening, you can collect Drosophila. So what you just need to do is you need to find some really fruity smelling thing, or a fruit, a cut fruit, ripened fruit kept in your kitchen, maybe somewhere. Even the peel of banana is good. You just keep it, you will see some flies coming around, and you can easily catch hold of those flies. And if you put them together, they will rapidly reproduce, and you will have large number of flies in your laboratory, only at your home also. So how much money does it cost? Nothing, 0 rupees, right? So you get a lot of flies in no cost. This is one. If you really want to authenticate your flies, then you have fly centers or fly bases across the country. In ISER is there, NCBS is there, Banaras Hindu University is there. You can get fly, right? So and one of my friends, Ashish Wani, in New Arts Commerce College, has brought some cultures from NCBS also. So the culture is not now available at New Arts Commerce College as well, and several mutants are also there. Another thing is, because you all know this fruit fly, like I said, it is called as tsatsan or tsatsni in Marathi. So you all know how small it is, right? So I will show few images. You can have hundreds of fly in one small vial, hundreds, fifties and hundreds of fly in one small bottle. Just like, those people who work microbiology, bacteria. So you on plate, you have 10 to the power 9, 10 to the power 12, 10 to the power 15 colonies. So similarly, in one vial, you can have a few hundred Drosophila at the same time, right? So it does not require a lot of space, and so handling is also not a problem, right? So you can have thousands of fly in one small lab. तो कोई बड़ी लैब भी नहीं बनानी पड़ती. If you go to some laboratories like BHU and ISER and NCBS, you will see that a 10 by 10 room is also sometimes enough to establish a Drosophila laboratory. No big deal. You can keep rest of the equipment outside the lab. But for culturing, a 10 by 10 room is also good enough. Even a smaller room is good enough. And like I said, say around 2 weeks is good enough generation time. So you get a large number of progeny, and therefore, because you have that progeny in only 2 weeks or so, you can do a lot of studies with respect to the phenotypes, with respect to the inheritance of genes, because you quickly get generations. No, F1 generation, 14 days, F2 generation, F3 generation, and so on, and you can repeat the experiments n number of times, because it is just a matter of 2 weeks or so. So several of my students, when they work on Drosophila, they find it very comfortable, especially for post-graduation studies, because they do not have much time with their studies also, with initial review of literature, and in the second semester again they have hardly 3 months or so. So they can quickly perform few experiments and come up with interesting results also. Now, another important thing is, if you put this Drosophila under a stereoscope microscope, then you will see the phenotypic features are very, very distinct, very, very distinguishable. Even the sexual dimorphism that you see, that means identification of male and female, it is also very, very easy. One can clearly look at all the parts of the fly, and therefore you can systematically define male, female, larvae, mutants, etc. So their identification is also very easy. Now, somebody was talking about the number of chromosomes initially when we were talking about the genome sequencing. When you have lesser number of chromosomes for genome, it becomes easier for us to carry out the sequencing of the genome. Here, there are only 4 pairs of chromosome, and you all know, I mean, several of you, especially from zoology, would know about the giant chromosome or the polytene chromosome. So there is this big chromosome where some of you would have made the slides also in your post-graduation or graduation studies using acetocarmine or acetorsine, where you see those bands, and puffs, Balbiani chromosomes, etc., the puffs, etc. Right. And another good thing is that even if you happen to release some of the Drosophila in your laboratory by chance or by accident, because these are not vectors for any disease causing pathogens, you are not worried about Drosophila, handling the Drosophila also. नहीं तो कई बार क्या होता है, आई एम डिलिबरेटली स्पीकिंग इन हिंदी बिकॉज देयर माइट बी फ्यू आउट ऑफ स्टेट स्टूडेंट्स। सो कई बार क्या होता है कि कोई फ्लाई है या कोई बैक्टीरिया है, कहीं कोई ग्राम नेगेटिव पैथोजेनिक तो नहीं है, कोई डिजीज तो कॉज हो जाए नहीं हो जाएगा, हम कुछ इनहेल तो नहीं कर लेंगे, या किसी और के साथ काम कर रहे हैं, किसी ऑर्गेनिज्म पे काम कर रहे हैं तो उससे कोई पैथोजन तो ट्रांसफर नहीं हो जाएगा, देयर आर कंसर्न्स, राइट? बट ड्रोसोफिला अच्छा गुडी गुडी ऑर्गेनिज्म, इट डज नॉट कॉज एनी डिजीज, डज नॉट स्प्रेड एनीथिंग, सो नॉर इट इज अ पेस्ट। तो अगर फैल भी गया तो ऐसा भी नहीं है कि उसकी बहुत उसकी वजह से कोई बड़ा नुकसान हो जाने वाला है, या कोई प्लाईवुड खराब हो जाने वाला है, या कोई इंस्ट्रूमेंट्स खराब हो जाने वाले हैं, नथिंग ऑफ दैट सॉर्ट्स। And a good thing is, again, as I said, in by year 2000, its genome also got completely sequenced, and 77% to be precise, and around 75% of the human disease genes are homologous in case of Drosophila. Now, there are very interesting studies. Sometimes I wonder, ki, if you look at dogs, they have certain behaviors. No, if you look at the face of dog, you will know if it is angry, if it is furious, if it is happy, if it wants care from you, if is looking for some adore from you, right? So similarly, I mean, Drosophila is a wonderful organism which shows a lot of behaviors in terms of getting attracted towards light, or moving away from the light causes a lot of impact on Drosophila. The geotropicism that you see in Drosophila, they move upward or they go down, and height, etc. So all these things matter a lot for Drosophila. So they have complex behavior. They have tremendous sense of smell also. I will give a list of some experiments which show that how wonderful this organism is. So they have tremendous sense of smell, not only in adults but also in larvae. They have some attractants, they have some repellents. They would be attracted in case of some chemicals, they would be repelled in some case of chemicals. So they have complex behaviors. And as I said, there are no ethical concerns. Nobody is asking you why are you carrying out Drosophila studies on Drosophila. Have you taken the ethical permissions or not? They are just flies. You can culture them in numbers. So mostly the permissions are not required. I carried it from Bangalore to my lab in flight. Nobody asks you why, and you can easily get permissions. Even Indian Speed Post allows you to post it and transfer or send it to some other lab. So Drosophila, it has very few ethical concerns also, and that is why I think Drosophila makes a wonderful model organism. See, all of you who are working on beetles, who are working on katla, and at some stage or the other, you will have to take permission of your ethical committee of your institute. But with Drosophila, you'll probably not, right? So there are several studies which have been carried out. I could not list a lot of studies which have been carried out on Drosophila melanogaster. Some of the areas that I mentioned is cancer-related studies that have been carried out. The latest being by the Mirzoyan in 2019, studies with respect to development in 2017, studies with respect to the nutrition in 2018, aging 2022, dipteran control with respect to pests. Now see, Drosophila itself is a dipteran, but can the studies on Drosophila be helpful for the control of dipteran vectors and pests? This has been studied by Zolfagari in 2015, and effect of insecticides and the development of resistance. This has been studied in one of the species of Drosophila, that is Drosophila Suzuki, as I was talking in 2019. So these are some of the recent studies which have been carried out in Drosophila. But Drosophila, like I said, in case of Saccharomyces, has won several Nobel Prizes, and for several reasons. Now, what is the reason that Drosophila is a wonderful choice? If you look at this image, which I have taken from Manchester Fly Facility. Now, fly facilities are places where you get Drosophila, several mutants, wild varieties also. So Manchester Fly Facility on their website has this image, and what it shows, you see, if you look at the color code. Now this color code has that gut part where this blue thing is there, then there is this red thing, and then there is this orange thing. If you look at this digestive tract of human being, so you would see same set of genes expressing in the foregut of human being, and then in the intestine, small intestine of the human being, and then the hind gut, that is large intestine of the human being. So that is the homology of the genes of the digestive system, right? How wonderful this is. Now again, if you look at the organization, body organization, so this cephalic organization of drosophila and the genes related to it is quite homologous to the cephalic organization in the human being. Same goes with the thoracic organization. Again, you can see the prothoracic, mesothoracic, and metathoracic getting distinguished in drosophila, and same way in human body as well. So that organization, even in the abdominal segments, the gene where they are expressed in the similar pattern, these genes get expressed in case of human beings also with respect to the body organization. So this color coding tells us exactly the same thing. And if you again go into the patterns of the circulatory system or excretory system, the muscles, the nervous system again here. So you see the nervous system here, and the nervous system in case of human beings here, you would see the same pattern. For almost every organs, there is a match in the fly. So that is why if you are carrying out studies on the Drosophila as a model organism, it is going to be a wonderful choice because it almost replicates everything in human beings with respect to the expression of the genes in the corresponding part of human being. Now similarly, if you would see, this is kidney in human being and the corresponding genes in excretory muscles over here, and the circulatory system over here, it functions in the same way, or the genes over there, they regulate the development, organization, and function of those organs in the same way in Drosophila. So what has that led to? इसकी वजह से क्या हुआ? Now इसकी वजह से ये हुआ. Now you see, we started Saccharomyces with 5 Nobel Prizes. Here you have several people starting from 1933 to 1946, 1995, 2004 to 2011, and then 2017. So Drosophila has got Nobel Prizes for how many? 6 years. 6 years on the trot, right? So 5 for Saccharomyces and 6 for Drosophila. So another wonderful choice as a model organism has been. So you have heard several of these names. T.H. Morgan, of course, you have heard. Then Mueller, you have heard. Naslin Volhard was one of the revolutionary lady who looked into Drosophila uh, from the modern perspective, right? And thereafter, it went on and on till Hall, Rosbach, and Young carried out their work. So now, when you are looking into any organism from the perspective of model organism, then you are interested that what all information is available. A lot of information is available online with respect to the Drosophila and the work which has been carried out. So there is this website which is called as Flybase, and just like we use the word database because this is a database of Drosophila, so we call it as a Flybase, right? The Flybase, it is online repository of the data, especially the genetic and molecular data of Drosophila melanogaster. So this is the screenshot of the Flybase website, where you can see here it has been written a database of Drosophila genes and genome. So just like you go and visit the NCBI site and look for genes, you look for PubMed, you look for BLAST, etc. So similarly, if you are specifically interested in Drosophila genes and genomes, you can immediately go into this database and look for the information that interests you. Because the link is there, I would like to show you that site as well. Is the site visible on your screen?
Yes, sir.
Right. So you have this site where you can do BLAST also. Then you have several other features like news and outreach, Drosophila journals and protocols, tools, meetings, courses, and jobs related to it, and how Drosophila is doing in disease research, interspecies work, etc. is going on. Then you can quickly search the homologs, gene groups, complexes, pathways, phenotypes, expression, whatnot, GAL4 related information also. So all these information is available on this database, and this database is commonly called as Flybase. So flybase.org is for people who are interested in Drosophila work. Another thing is Flight Book, right? Now, this is a journal published by Genetics Society of America, and it provides the information of the latest upcoming things in Drosophila genetics. So those who are interested in Drosophila genetics, they can go to this website. If you can see on your website, can you see? I hope you can see. I can see that I have shared this,
Yes, yes.
right? Yes. So you see, if you really are interested in any aspect of Drosophila, right, from cell signaling to development, ecology, gene expression, genome organization, method, protocols, DNA repair, recombination, stem cells, germ lines, etc., all the information is available on this particular page, which is termed as Fly Book, right? So it talks about the genetics of Drosophila and is published by Genetics Society of America. Another interesting thing. So the good thing with Drosophila, as I said now, is that you have a lot of resources available with this. Now, this is Bloomington Drosophila Stock Center. So if you want to have Drosophila, you can get it from BDSC. The website is bdsc.indiana.edu. But why go to Indiana? Indiana में जाने की जरूरत क्या है जब इंडिया में अवेलेबल है? So if you want several cultures of Drosophila, you can contact NCBS, that is National Center for Biological Sciences in Bangalore. You can also contact ICER in Pune. ICER will help you in establishing a Drosophila lab if you want to establish one. Department of Zoology at Banaras Hindu University has also a Drosophila facility. And as I said, even New Arts Commerce College, Dr. Ashish Wani is having some strains now, which I hope he has subcultured. So you can get the cultures from there also, right? So all the resources which are available to study Drosophila and get Drosophila are readily available. And there is good amount of literature which is available also. If you want, I can share the Google Drive link later on with the information available for Drosophila. Now, just to increase your attention and interest in Drosophila, how they can be interesting, I'll share a few images and slides which will probably um, take your attention towards how Drosophila can be an interesting aspect for your research. Okay, so it is a diptera insects which shows sexual dimorphism and a prominent one, right? So you can easily identify male and female. Saraji, if you can please mute yourself, your microphone is unmuted. Thank you. Sarah, Sarah ma'am, thank you. Okay. So the top three are the females. Now, if one can tell me, how do you think this one, that means A, is different from D? Can somebody tell me how A is different from D? Forget the rest of the others.
Abdomen size broader, narrower.
Correct, correct, correct. Very nice. So in the this image, the A1, here you can see that abdomen is pointed, right, tapering, and comparatively, the may this one has a rounded abdomen and is quite smaller, right? So several segments they merge. The posterior segments they merge together, and in this one, the posterior segments are quite clear, and the end is quite pointed, right? So this is female, right? And from the ventral side, you can observe the female here. and this is from the ventral side, male's abdomen, right? So you will see that females are comparatively larger, bigger, and males are comparatively shorter. Male will have a rounded abdomen, female will have pointed abdomen, male will have some segments which are fused, and females will have distinguished segments, the posterior segments. But another important aspect in identifying a male and female is this thing which you will see in the prothoracic pair of leg, and this is called as sex comb, right? So here they will have this, if this, you can observe this black spot over here in the prothoracic leg, that is called as sex comb. So when you can identify a male and female morphologically, you call it a sexual dimorphism. So this is the sexual dimorphism that you see not only in adults of Drosophila but also at the larval stage. Now see here in the image, you can quickly observe that this is the pupa of a Drosophila. Now this is the female pupa and this is the male pupa. Even in the pupal stage, you can see here these two black spots, which clearly tell us that this is a male pupa. So you need not wait for the pupa to emerge, the fly to emerge, right? Even at the embryonic stage, in the pupal stage, you can identify whether it is a male or whether it is a female. So that actually will help you to separate the male and female even at the embryonic levels. This is one advantage with Drosophila, working with Drosophila also. Now then, the question that comes to my mind, okay, fine, I'll be working on Drosophila. How expensive can it be? So what do you require? You require these petri plates. So any company like Tarson or Laxpro will probably offer you a petri plate of, say, for example, 85 size. So PD 85, PD 100 will do. PD 100, PD means petri dish, right? You can have these vials, small vials or bottles. We sometimes use those Rajhans ka doodh bottle or Amul ka doodh bottle also. They also work fine. No problem. The only thing is you need to learn the art of autoclaving, working under aseptic conditions. So that is the only thing. Then what is this thing? Dal chawal kya rakha hai? तो दाल चावल भी बड़ा सस्ता है Drosophila का. You need agar. Bacteriological agar is fine with this. और कितना तो लगा, लगभग 400 ml पानी में लगभग 3 ग्राम 2 ग्राम agar. So that is fine enough. Maize powder, corn flour, जो हम सोयाबीन चिली में लगा करके खाते हैं, that corn flour, sugar, sucrose you can use. There are few media where jaggery गुड़ को यूज करते हैं. ईस्ट वी ऑल गेट इन द मार्केट. बाजार में जाओगे इडली में डालने के लिए या ब्रेड बनाने के लिए या केक बनाने के लिए ईस्ट मिल जाता है. सो यू कैन यूज ड्राइड ईस्ट. और 1 ग्राम और 1 ml दो चीजें लगती हैं. एक मेथिल पैराबेंजोएट which is also called as nipagin, methyl paraben भी बोलते हैं उसको, and propionic acid. So they work as antifungal, right? तो वो एंटीफंगल करके काम करते हैं. Therefore इसमें और कुछ फंगस ग्रो नहीं होती. Only Drosophila grows. So it costs nothing, almost nothing, right? ये लगभग 500 ml 400 ml बना दिया. 400 ml में तो कम से कम ऐसी 15-20 वॉयल्स भर जाएंगी और ऐसे 6-7 बॉटल्स भर जाते हैं, right? और ऐसी बहुत सारी प्लेट्स भर जाते हैं. So you can culture hundreds of Drosophila in this much, right? So the point that I'm trying to make is it is hardly expensive. You can always work on Drosophila if you are running short or running tight on your pocket. And what instruments do you usually need to study Drosophila? A stereo microscope like this, बहुत अच्छा. थोड़ा सा उसमें zoom चाहिए, थोड़ा light अच्छा चाहिए, ऊपर से light चाहिए, थोड़ा नीचे से light चाहिए. That's it. So a stereo microscope of good quality. और कोई बहुत बड़ा कंपनी भी नहीं चाहिए, ठीक ठाक कंपनी का रहा तो भी चल जाता है. अच्छा, okay. So for routine Drosophila work, what equipment or instrument you would be using? Small equipment like you would be using some syringes and needles, forceps, some brushes, some slides, some cavity slides, some droppers. Morgue. Morgue means the Drosophila which you have worked on, you would put them into morgue. Morgue means होता है, morgue means जहाँ पर dead body रखते हैं. So those etherized Drosophila which you have worked on, if you want to discard those flies, you can discard those flies into this thing. Then you would need ether, diethyl ether to anesthetize the flies. 70% ethanol to transfer the flies. Sometimes if you want to observe those larvae, say for example, then you would need 70% ethanol and ether. So that's it. This is an etherizer. And etherizer can even be simpler. You can take small funnel and have cotton around the nose of that small funnel and put few drops of ether, and when you transfer the flies, due to that ether, the flies get anesthetized. You can easily observe them under the stereo microscope and dissect them out, look at the larva, observe the mutants, and do whatever you want, right? Several experiments. So if you have this basic arrangement in your laboratory, you are good to go with the basic Drosophila work in your laboratory alone. So somebody was asking me the other day, "Sir, do you have a Drosophila lab?" I said, "Lagta hi kitna hai," right? So there's not much that we require to establish a Drosophila lab. Now then we come down to, I hope I'm still there. Can you all hear me?
Yes, sir.
Yeah, yeah,
Yes,
fine,
sir.
sorry. Yeah. So now these are some of the interesting images with respect to the Drosophila. As I mentioned, that one of the qualities of model organism is they can have several mutants. We saw that in case of C. elegans also, and here Drosophila is famous for n number of mutants. And some of the mutants which for which the pictures have been given here are with respect to the eye. Now you can see here, this is a white eye mutant. The normally wild Drosophila that you see has a red eye, right? But the Drosophila that you see here, you see this is the wild type Drosophila. So beautiful red eyes are there. Now if you go here, you see that this is a white eye mutant. So depending upon what phenotypic aspect has been affected, you call them as the that particular mutant. So this is white eye mutant. Then there is white apricot eye mutant. Then this is brown eyed mutant, rosy eye mutant. Now this sometimes is difficult because there are only few little variations in the color. So though it is very easy for me to say over here, but you see very small variations in the color. This is scarlet, slightly bright, scarlet eye mutant. Then this is claret eye mutant. This is sepia. This is quite identifiable. This is sepia eye mutant. Then this is bar eye, just like a rod. So bar eye mutant. So all of these are the mutants with respect to the phenotypic changes in the eye. And in bracket, what you see is this WA, W, BW minus, RY, ST, CA, SE, BFI. All these are actually the genes which got mutated. The symbol of the genes are also there. Then there are some mutants where the body color changes. So here is one mutant where the body color is yellow. So you call this as yellow body mutant. Then here it is, you call it as, it is quite darker. You call it as ebony. Then here you see that the wings have shortened. So you call them as vestigial wing mutant. And here the wings have got curl. So this is called curled wing mutant. Now, uh, here also there is slight variation. One more mutation that we'll see, it is called as curlio. So both look similar almost to certain extent, but there is a small variation that you will see in future. You see this curly wing and with bristles. So this and this is sepia eye colored with curled wing. So curly wing and curled wing are two different varieties of mutations. So here you see that there are vestigial wings also, but sepia color eye also. So double mutant. This is called as double mutant. Here also you have sepia colored eye and curled wings. So this is also a double mutant. This is, see, this is gene CU and this is gene CY. So there are two different genes. This is curled wing and this is curly wing. So this is curly wing, and then there are bristles also. You can see here bristles, right? So the bristles mutation is also there. Then this is yellow body and white eye and forked bristle. The bristles, if you see under the microscope, they will be forked, right? So this is triple mutant. There can be multiple mutants also. So raphoid eye, curled wing, claret color, stripe, ebony, all together. So you can have multiple mutations as well. Now, uh, You can, to get aware of how Drosophila functions, can perform several experiments in your laboratory only. I could see that there was one student in the chat box who is probably working on Drosophila. So you can start your work with the rearing and handling of the Drosophila. You can bring Drosophila into lab, make culture medium, start growing it, get a feel of it, observe it under the microscope, let them breed, observe the eggs, then let the larvae come out, then pick up first instar, second instar, third instar larva, observe those larvae under the microscope. Then if you have come across some mutants, you have some mutants in your laboratory, or if you can order some mutants, then go and look into some mutants, their phenotypes also, try to observe them. Then you can solve some problems also, and not only solve some problems, but also allow some mutants to crossbreed together and see if there is some Mendelian inheritance taking place or not, or whether linkage is taking place or not, whether 2-point linkage is taking place or not, right? So there are several possibilities with respect to the genetic crosses as well, and based on that you can analyze the recombinant frequencies from a trihybrid cross and calculate the genetic distances as well. Then heat stress tolerance in Drosophila. As I said, the Drosophila does well at a temperature at say around 25 degrees Celsius. So what happens if you increase the temperature, say to example 30 degree, then to 35 degree, then to 40 degree? Do they faint? Then if they faint, how much time do they require to revive again? Similarly, if you go bring it down to say 15 degrees and 10 degrees, and correlate this with the time duration for which the temperature has been increased or decreased, you will see that there are different responses. So there are temperature responses as well, and like I was telling you, the olfactory responses. The larvae are extraordinary in identifying some chemicals and moving only towards. So you can perform plate assays where on the plate you will have agar along with say some yeast, and then release the larvae at the center, and you can have say attractants on one side and repellent on one side, and you will see that larvae easily move towards the attractant, some of the chemicals which Drosophila prefers. Then there is another study which we call as negative climbing assay. Climbing assay is where like I said, Drosophila, no, though they prefer to move upward, right? If you put them in vial, they will always be going upward. So if you bang them on the floor, they will start climbing up. But if you keep